Hello my dear students. Today I am here with the topic cholinergic neurotransmitters. This is another unit that we have to study. We have already discussed about adrenergic agents, non-adrenergic agents, adrenergic neurotransmitters, right? So now in this video today I'll try to explain about cholinergic neurotransmitters and different cholinergic agents. Alright, so what are cholinergic agents? First thing that you need to recall is that autonomic nervous system is of two types, or it is divided into two parts, that is sympathetic nervous system and parasympathetic nervous system. Sympathetic nervous system, if you remember well, if you have studied the previous presentations that I gave you, sympathetic nervous system, basically we are talking about adrenergic nervous system, adrenergic system, right? And when we are talking about sympathetic nervous system, that means we are talking about cholinergic systems. Okay, so parasympathetic nervous system is also known as cholinergic system. And why is it known as cholinergic system? Because it uses an enzyme which is known as acetylcholine. When we are talking about an enzyme named acetylcholine, then we are discussing about the cholinergic system. Alright, so in another word, cholinergic system is one in which acetylcholine is involved. It is used or released in the body. Okay, few points about acetylcholine. First thing, that it propagates impulse transmission in the parasympathetic nervous system. Where is it present? It is present at postganglionic synapse of parasympathetic nerve endings. Okay, so they are present at the parasympathetic nerve endings. Okay, this is the structure of acetylcholine. If you see, this part is the acetyl part, right? CH3, this is considered as CH3, right? CH3COO, right? So acetyl part, this is, remember acetic acid formula, CH3COOH. So what you have here, CH3COO, right? H has been removed, so acetic acid becomes acetyl. Okay, and this full part is known as, this structure is the structure of choline. Alright, so this is the structure of acetylcholine. What else you can see in here is, this is an ester, right? General formula for ester is R, then C. I know I'm going slow, please bear with me. RCOO R, right? This is the general formula for ester. Here you can see here that it is RCOR, R, CO, O and R, right? So this is a type of ester also. Plus this is having a plus charge, that means the nitrogen here is quaternary, right? Alright, so acetylcholine will act in the brain and in the body of human beings and animals as a type of neurotransmitter. Previous adrenergic system neurotransmitters, I hope you remember. Okay, then for a cholinergic nervous system, the neurotransmitter is acetylcholine. For adrenergic nervous system, if you remember, it was epinephrine, norepinephrine, right? Okay, so acetylcholine will act in the brain and body of humans and animals as a neurotransmitter. Now, as you can see here, the structure consists of ester, acetic acid and choline. Right now I just explained you the structure, that this part full is acetic acid, acetyl part, so acetic acid. Then choline means this structure here, if you can compare the two, see till here full same, right? This part, so choline part, right? And ester, as I told you, RCOOR, so this part is ester. Hence the structure of acetylcholine consists of ester, acetic acid and choline. Alright, this. Now, biosynthesis of acetylcholine. Please focus and try to understand. This is serine. Okay, serine in the presence of enzyme serine decarboxylase. Again, decarboxylase. As soon as I say decarboxylase, immediately it should strike you that there is a carboxylic acid group that has to be removed. Decarboxylation has to take place. So this will be removed as specified by the name of the enzyme itself. What we are left with is OH, CH2, then CH2 and NH2. Okay, because once OH acid has been removed, one hydrogen will come here. So from serine you get ethanolamine. Serine was having a carboxylic acid, so this carboxylic acid group was removed with the help of enzyme serine decarboxylase, and from serine we got ethanolamine. Okay, then this ethanolamine is converted into choline. Ethanolamine is converted into choline in the presence of enzyme S-adenosylmethionine. Alright, so ethanolamine is converted into choline in the presence of enzyme S-adenosylmethionine. As you can see in here, OH, then this CH2, then this CH2 and this nitrogen. Okay, this NH2 group, what will happen? The two hydrogens will be removed and 2 methyl groups and one more methyl group will be here, and this will become plus. Alright, nitrogen is now having 1, 2, 3 and 1, 4 valencies, right? 3 to the methyl groups and 1 to this CH group, right? So now since carbon has got nitrogen has got 4 valencies, so nitrogen will have a plus charge. This is choline. Okay, this part has taken place in liver till choline. Okay, choline synthesized in liver. Alright, then choline will go in cytoplasm, and in the presence of enzyme choline acetyl transferase, again the name, focus, choline acetyl transferase. That means at the choline, in the choline moiety we are going to add an acetyl group. We are going to transfer an acetyl group. Now from where will this acetyl group come? It will come from acetyl coenzyme A. Okay, it will come from acetyl coenzyme A. And from where is this acetyl coenzyme A coming? It is coming from pyruvate. Pyruvate in the presence of pyruvate dehydrogenase will convert into acetyl coenzyme A. This part, conversion of acetyl coenzyme A, formation of acetyl coenzyme A is taking place in mitochondria of nerve terminals. Okay, this is not mitochondria, it is mitochondria. Please correct it when you make the notes. So pyruvate converts into acetyl coenzyme A in the presence of enzyme pyruvate dehydrogenase. This conversion is taking place in the mitochondria of nerve terminals. Okay, now this acetyl coenzyme A will transfer the acetyl group to the choline here in the presence of enzyme choline acetyl transferase. So what is going to happen in here is this hydrogen will be removed. Okay, remaining is same here. You can see the nitrogen with 3 methyl groups and 1 CH2 here, then again CH2 and then O. This O we are talking about, and this is the acetyl group. Okay, so acetyl group is being transferred. From here this oxygen, this full will be removed, OH group, and this acetyl moiety will be added to it. Okay, CH3COO. So this makes acetylcholine plus coenzyme A. The acetyl coenzyme A will then be converted into coenzyme A, right? Okay, so this was about biosynthesis of acetylcholine. Once again I'll go through in short. Serine will be converted into ethanolamine in the presence of enzyme serine decarboxylase. That means there is an acid that has to be removed. Decarboxylation has to take place. So this acid will be removed and the remaining structure remains the same. OH, CH2, CH2, NH2. Right, this carbon will get free valency, but it will get when COH is removed, this carbon will get a free valency and it will satisfy itself by a hydrogen. So this becomes, instead of CH, it becomes CH2 here. Okay, then this ethanolamine is converted into choline. It is converted into choline in the presence of enzyme S-adenosylmethionine. Alright, in this step what is going to happen? The hydrogen here with the NH2 hydrogens will be removed and methyl groups will come here. Methyl groups have come here, 3 methyl groups and 1 here. Since nitrogen got 4 valencies, so we have a plus charge here. Okay, this full from serine to choline has been done in liver. Alright, then choline comes in cytoplasm, and there this choline is converted into acetylcholine in the presence of enzyme choline acetyl transferase, and the acetyl moiety that has to be transferred to this choline is achieved from acetyl coenzyme A, which is synthesized in mitochondria of nerve terminals from pyruvate in the presence of enzyme pyruvate dehydrogenase. Okay, so you get acetyl choline plus coenzyme A. This is taking place in cholinergic nerve endings. Okay, choline to acetylcholine is taking place in cholinergic nerve endings. Acetylcholine, again as it's written here, acetylcholine in cholinergic nerve endings, that is what I have written here. This is acetylcholine is synthesized in cholinergic neurons or cholinergic nerve endings. Then where is it stored? It is stored in the cytoplasm. In cytoplasm, where? In small synaptic vesicles. Okay, when these vesicles contain cytoplasm, these vesicles contain acetylcholine, and when this acetylcholine has to be removed out or moved out in the open in the vicinity, so what is going to happen? It is excreted by exocytosis. Okay, I hope you all know what is exocytosis. Alright, so with exocytosis, what is going to happen? It will be excreted. When I say excreted, it means it will be expelled out. It will be expelled out by exocytosis. Alright, so it is stored in the cytoplasm in small synaptic vesicles. Alright, and excreted by exocytosis. Next is metabolism of acetylcholine. The functions and effects of acetylcholine can be stopped or inhibited by enzymatic hydrolysis. Okay, which enzyme does this? The enzyme is known as cholinesterase or acetylcholine esterase. Okay, same only, cholinesterase or acetylcholine esterase. Alright, so to inhibit or to stop the action of choline, right, what can be done? Enzymatic hydrolysis. Which enzyme is involved for this? Cholinesterase or acetylcholine esterases. See here, this is the structure of acetylcholine, right? This part, choline part, and this is the CH3COO, acetyl part. So acetylcholine in the presence of enzyme acetylcholine esterases is metabolized, and you get one acetate ion here, CH3COO minus, and one choline over here. Okay, this is choline. So for metabolism of acetylcholine, simple it is, acetylcholine will be metabolized into acetate ion and choline. Alright, this choline again will be reuptaken into the terminal for again further synthesis of acetylcholine. Okay, this is about metabolism of acetylcholine. Now, cholinergic receptors. Cholinergic receptors means acetylcholine will go and bind to some receptor, then only it will give the actions, right? So what are the receptors? Cholinergic receptors, there are two different types of receptors to which acetylcholine will bind. First is muscarinic receptor or M receptors, and the other is nicotinic receptor or N receptors. Okay, two types of receptors, muscarinic and nicotinic. Now see, muscarinic receptors are five types: M1 or neural type, M2 cardiac type, M3 glandular, M4 and M5. So these are the five types of M receptors. Then N receptors then are of two types, NM and NN. NM is muscular type and NN is neuronal type. These I have given you the names and different types of N receptors and M receptors, that is nicotinic receptors and muscarinic receptors. You should go through, your notes will be provided to you. I want you to go through the function and location of all these. Okay. Now, parasympathomimetic agents. Mimetic means mimicking, similar. So when I say parasympathomimetic agents, that means those compounds which will mimic the action of acetylcholine. Okay, so that means they will have the similar action as that of acetylcholine. Now, parasympathomimetic agents are of following types. First, cholinesterase inhibitors. Cholinesterase, I hope you remember the enzyme that was terminating the effect of acetylcholine. How it was terminating the effect of acetylcholine? It was metabolizing it into acetyl and choline. Just now, here only you saw, right? The enzyme is cholinesterases. So, cholinesterase inhibitors will stop the metabolism of acetylcholine. So, if acetylcholine is not metabolized, that means it is more available in the body. Second is direct acting agents, and the third is cholinesterase reactivator. Alright, we'll discuss about them. Then first thing that you need to know is that cholinesterase inhibitor is again of two types. First is reversible direct acting, and then is irreversible indirect acting. Okay, so this is the type of parasympathomimetic agents. Cholinesterase inhibitors, which are of two types: reversible direct acting, irreversible direct acting. Then second is direct acting agents, and third is cholinesterase reactivator. Examples of them: direct acting agents, for example acetylcholine of course itself, carbachol, pilocarpine. Then reversible direct acting agents can be physostigmine, neostigmine, pyridostigmine, tacrin. Okay. Then irreversible indirect acting will be malathion, parathion, and cholinesterase reactivator is pralidoxime chloride. Okay, these are the different examples of the classification that we just saw. Alright, so this is the classification of parasympathomimetic agents, and these are some examples of the same. Okay, next class we will discuss the SAR of parasympathomimetic agents. So this was all for this video. Okay, I hope you have understood the basic concept of cholinergic agents. You know which is the cholinergic neurotransmitter, that is acetylcholine, right? You should know what are the functions of acetylcholine in short. Then where it is stored in the cytoplasm, in the vesicles, right? And then when it has to come out, it will be expelled from the vesicles through exocytosis. Okay, so you should know the storage, you should know the metabolism, you should know the synthesis. That is all I have discussed. Plus I have discussed the classification of sympathomimetic agents, and the receptors also I have discussed with you. Okay, so in case of any doubts, please let me know. I hope you have understood this much. Alright, thank you very much class.